Good morning to one and all present here. I am B.Priyadharshini, Assistant Professor, Department of Biotechnology in KSR College of Arts and Science. Today I am going to present you an interesting topic which is known as in vitro fertilization. In vitro, in the name itself you can identify, that means in laboratory the fertilization occurs. Why in laboratory fertilization is important means when there is a cause of infertility in male or female, they will be going for this in vitro fertilization. Next. Definition: in vitro fertilization is a process of fertilization where an egg is combined with the sperm in laboratory, that is in vitro. The process involves monitoring and stimulating a woman's ovulatory process, removing an ovum or ova from their ovaries and letting sperms fertilize them in a cultural medium in a laboratory. Next. In vitro fertilization maximally involves 5 steps. First one is ovarian stimulation, which is known as hormonal therapy. Second one is egg pickup, which is known as follicular aspiration. Third one is sperm preparation. Fourth one is egg fertilization. Fifth one, embryo development. Then finally we are going to transfer the embryo through the cannula. Next. There are 5 basic steps to in vitro fertilization. Step 1, stimulation. This is also called as superovulation. Medicines called fertility drugs are given to the woman to boost their egg production. Normally a woman produces 1 egg per month. When fertility drugs are given to the woman, the ovaries will produce several eggs. During this step, the woman will have regular transvaginal ultrasounds to examine the ovaries and blood test to check the hormonal levels. These are the tests first undergone to the woman. Next. This is this transvaginal ultrasonography. So, here abdominal transducer is there and vaginal transducer is there. By using this ultrasound only, the follicles are viewed. Next. Egg retrieval. Step 2 is egg retrieval. A minor surgery called follicular aspiration is done to remove the eggs from the woman's body. This surgery is done by the doctor in the hospital. The woman will be given medicines so that she doesn't feel the pain during the procedure. Using ultrasound images as a guide, the healthcare provider insert a thin needle through the vagina into the ovary, that is where these follicles are present containing the eggs. The needle is connected to a suction device which pulls the eggs and fluid out of each follicle one at a time. When the procedure is over, they will be further moving to the next ovary. There may be some cramping after the procedure, but it will go away within a day. In rare cases, a pelvic laparoscopy may be needed to remove the eggs. If a woman does not or cannot produce egg, they can move for the donor eggs. Next. So, this is the process. See, the ovarian stimulation occurs. They are giving the fertility drugs to the woman. Next, it is monitored by using the transvaginal ultrasonography. Okay, the follicular size are repeatedly checked for them. Then, this is the method how the eggs are retrieved from the ovary. This is the follicular aspiration. Here the oocyte is taken with the help of follicular aspiration and the oocyte is taken in the culture medium in the laboratory. Next. So, step 3 is insemination and fertilization. Insemination is the mixing of sperm and egg. So, the man's sperm is placed together with the best quality eggs. The mixing of the sperm and egg is called insemination. Eggs and sperm are then stored in environmentally controlled chamber, which is known as incubator. The sperm most often enters an egg after a few hours of insemination. So, it takes time to fertilize the egg. If the doctor thinks the chance of fertilization is low, the sperm may be directly injected into the egg. When the sperm quality is very low or sperm rapidity is very low, the doctor will choose the decision that they can go for ICSI. ICSI is nothing but intracytoplasmic sperm injection. Here the sperm is directly injected towards the egg. Many fertility programs routinely do ICSI on the some eggs even if they appear normal, because the rate of success in ICSI is more than the rate of success in IVF. Next. For step 4 is embryo culture. This is a very important step. When the embryo fertilized, it started dividing. Okay, it becomes an embryo. The laboratory staff will be continuously monitoring the growth of the embryo. That is, the cell division is monitored by them. Within about 5 days, a normal embryo has several cells that are actively dividing. Couples who have high risk of passing genetic disorder, that is hereditary disorder, to a child may consider pre-implantation genetic diagnosis, which is known as PGD. The procedure is most often done 3 to 5 days after fertilization. Here, the laboratory scientist will remove a single cell from the embryo and they will be screening it for the specific genetic disorders. According to the American Society for Reproductive Medicine, PGD can help parents to decide which embryos to implant. This decreases the chance of passing a disorder onto the child. So, if any genetic disorder is there, they can view it by the process called PGD, pre-implantation genetic diagnosis. That is, pre-implantation means before implantation, implanting the embryo to the woman, they can screen it and they can check it, and if any genetic disorder is there, they will not implant that embryo into the woman's womb. So, this is very important process. This decreases the chance of passing a disorder onto a child. The technique is controversial and it is not offered at all the centers. So, you can see the picture. See, in day 1 you can see the zygote, okay, 2 cell stage. Next, in day 2 embryo is in 4 cell stage, day 3 8 cell stage, then it forms morula, in day 5 blastocyst. So, cell divides rapidly. Next. Next, embryo transfer is the 5th step. Here, embryos are placed into the woman's womb 3 to 5 days after egg retrieval and fertilization. That means they will place it in a morula stage or blastocyst stage. The procedure is done in the hospital. The doctors insert a thin tube. This is known as the thin tube, which is known as embryo transfer catheter, okay, containing the embryos into the woman's vagina through the cervix and up to into the womb. If an embryo sticks to the lining of the womb and grows, pregnancy results. That is known as implantation. When the embryo sticks to the lining of the womb, the process is called as implantation. More than one embryo may be placed into the womb at the same time, which can lead to twins, triplets or more. So, depending upon the quality of embryo, the embryos may be placed inside the woman's womb. But here is a factor dependence is there, which is known as woman age. The exact number of embryos transferred is a complex issue that depends on many factors, especially the woman's age. You can see a syringe is there, this is known as catheter, and a medium is there, and here they are placing the 3 embryos into the woman's womb. So, this is known as a embryo transfer catheter. Next. So, unused embryos may be frozen. This process is known as cryopreservation. They are storing the unused embryos in the liquid nitrogen by using a cryoprotectant. Then the process is called as cryopreservation, and it may be implanted or donated at a later date. Whether if the implantation fails for the particular couple, they will be using the frozen embryos, or they will be donating their embryos to some mother to get fertility for them. Students, I hope you understood what is in vitro fertilization. So, there are 2 major points you should remember here. Here I have told you what is in vitro fertilization and what is ICSI, which is intracytoplasmic sperm injection. Okay? Then the important terms you should know here is cryopreservation, ovarian stimulation, then follicular aspiration, embryo transfer. You should know the role of catheter also. Thank you.